All right. So, yeah, I'm Norman. I'm connecting from Berlin in Germany. And yeah, just first a few words about our company called Scalable Minds. We're based in Germany, right next to Berlin, and we've been around for quite a while now. And we work with a number of different institutions from all over the world. What really motivates us is a chart like this. So here you can see a few data sets that have been published in recent years in the field of neuroscience. These have been published already. These are some projects that people are working on, and of course there's a bit of speculation because this axis here, the time axis, goes well into the future. Nevertheless, there's a trend that we can observe here, which is if you look at the axis over here, you can see that there's actually a logarithmic axis. So the sizes of data sets that have been produced and are projected to be produced are growing exponentially. And that facilitates a new way of working with these data sets from a software perspective, from storage systems and compute systems that are required to work with that. And there's another trend, of course, and that's AI. Everybody's talking about it, but today AI is already a quite mature tool for all sorts of bioimage analysis tasks. But still, it can be quite complicated to use for biologists, sometimes even bioimage analysis. So we believe that there is a way to make all of this more accessible to more people. And then there's another trend, which is that data sharing and collaboration among scientists is really increasing. And there are a number of different reasons for that. One reason is that these data sets that are being acquired today are just so big and rich that you need a multidisciplinary team to make use of them and to analyze that thoroughly. Also, there's some demands from journals and funders that the data is being shared. Yeah, and more often than not, the experts that you want to work with, they're not in the same room or not even in the same country, but they're spread around the globe. So software is also required for that to make that easier and to enable this level of collaboration. So with that, I want to introduce you to Webknossos, which is a web-based platform for doing collaborative projects on very large images. And we developed that together in collaboration with the Max Planck Institute for Brain Research. So Webknossos has a bunch of different features. I'll go through of them in this presentation right now, and I'll also later on have some short demos that I can show you. The primary feature really is visualization of very large data sets. So this one is a 50 terabyte data set of a full fly brain. It's been acquired at Janelia a few years ago and published. And you can interact with these data sets directly from a web browser over the internet. So you don't need to download any data, you don't need to download any additional software. You can just click on a link that somebody might send you or that you find in the paper, and then you can start exploring these data sets. You can zoom in and out, you can move around, and again, everything just runs in a web browser. And then if you found something interesting, then you can also just take the link and send it to somebody else, and they will also be able to look at that, right? So sharing and collaboration is really easy because everything runs inside of a web browser. So I'll have a lot of electron microscopy data set in my talk, but Webknossos also works great for a number of other modalities as well. Here you can see a light sheet data set or a light sheet movie that has multiple time points, so you can move around in time as well. And I'll talk about annotations later, but you can also use all of the annotation tools that Webknossos has as well. So it's not only great for EM, it's also great for X-rays and all sorts of light microscopy data sets as well. A new feature that is coming and that we're working on for this year is to better support correlative imaging approaches. There's already a feature where you can overlay multiple image modalities in Webknossos and look at them, and we're working on registration features as well so that you can register these images together. And that works together with a number of under the hood uses different transformation types. So affine transformations are well supported, but also nonlinear transforms like spline transforms are also supported. So today, if you know the tool called BigWarp, then you can already import BigWarp files into Webknossos and then get some transforms out. The example that you're seeing here are two X-ray images laid on top of each other. One has been acquired by micro CT in a lab, which is lower resolution, and then the other one is a synchrotron X-ray image that has been acquired, a synchrotron, and is much higher resolution, and they're overlaid on top of each other so that you can look at both at the same time, and then basically trace also across the scales. Webknossos is not just a visualization tool, but it's also a tool where you can store and manage data sets all in one place. So you can invite others into your account, and then you can put all your data set in there. You can organize these data sets by folders. You can attach tags to them. You can attach arbitrary metadata to that. Also, you can attach permissions to that. So if you run a facility, then you can have one folder per project, and then just assign the people that need to have access to that data set to then work with that. And then they can just open up their browser and they will find all the data that belongs to their project right inside of Webknossos. We tried to model this similar to how Google Drive works like. So if you work with that before, then you will feel right at home. So the second big category of features that Webknossos has is annotation tools. And there are a bunch of different annotation tools inside of Webknossos. And the first one that we're looking at here is a segmentation tool. It's actually a semi-automatic segmentation tool, which is powered by the Segment Anything model that has been published by Meta AI a few years ago. We're using version 2, which also has support for, or enables us to implement like 3D segmentation. So with a single click, you can segment out a number of different sections. Segment Anything actually uses this for tracking objects in videos, and we're using this for segmenting 3D, and it works really great for a number of different biological features, which can be a bit surprising because the model is actually a generalist model, has been trained on cats and dogs and cars and airplanes and all of these things that we have to click on different websites. So, but it also works great for this bioimaging data here, and can speed up the process of segmentation quite a bit. And again, everything runs in a web browser. You don't need to install any plugins or anything, and you can also collaborate with other people on these things. You can just send them a link, and then they'll be able to help you with the annotation work. Because everything runs in a web browser, you can also use Webknossos wherever a web browser runs. Here you can see an iPad, which is nice because then you can use the pen to do very precise edits on the segmentation if you're looking for the highest quality, but also if you just want to have this direct manipulation of the data, which is quite a nice workflow. Once you've done all the segmentation work, then you can also get statistics out from Webknossos. So there are statistics like the volume of the segments or surface areas that are in here, and we'll add more types of statistics. So if you have any suggestions of what would be most useful for you, then please let me know, and then we can include that in future releases of the software. Other things that you can do once you have created some segmentations is you can visualize them as 3D meshes, and you can visualize them inside of Webknossos, but you can also export them and then load them into Blender or another animation tool for making nice figures or animations. Webknossos also has a feature for rendering out animations directly built in. So if you want a quick animation for a lab presentation or something like that, then you can also use the built-in features of Webknossos to generate these animations. If you're working with connectomics data, then there are a few specialized features inside of Webknossos that make it easier to edit automated segmentations. So there's a proofreading feature that allows you to do split and merge operations quite efficiently, and you can do that either in the EM data or on the meshes. So it's a nice way to basically correct any mistakes that the AI systems have been doing. But this is a specialized feature for connectomics analyses. And because everything in Webknossos runs in the browser, it actually runs on a server, and the browser talks to an API to the server to get all the data and do all the work. And the nice thing about that architecture is that you can also talk to the server directly through an API. And to make that a bit easier, we have published this Python library, which allows you to access data on Webknossos through a programmatic interface. So here in this short example, first there's a Webknossos context that you open, and every user has their own token for authentication purposes. Then once you're authenticated, then you can access datasets in Webknossos. You can access the individual layers that this dataset might have if there are multiple ones, and then you can also access this MAG, which is like the resolution level. And once you have that, then you can actually read out some data in there. And with that data, you can then run any analysis that you want through that might be available in your Python toolbox. And so this is a very short example. There are many more functions. You can also upload data to Webknossos, and you can automate a lot of workflows for importing data sets into Webknossos, attaching metadata to them, downloading them. So a workflow that some of our users like to do is to download some raw data from Webknossos, then do their own segmentation or analysis on top of that, and then upload the result, like a segmentation layer, back to Webknossos for visualization and for sharing so that they can get the feedback from their colleagues or collaborators on that. So that's something that can be facilitated through this library quite nicely. And so under the hood, this library makes extensive use of Zarr, and which brings me to the next section in my talk, which is talking about OME-Zarr and next generation file format. So I'm sure many of you have heard about this file format already. If not, then I'll give you a brief introduction to what it actually is and what it can help you with. So just imagine that you have a data set that might look like this. So this is your sample, and then if you put it into a microscope, let's say an electron microscope, then it will be acquired section by section, and your microscope will produce like a single image file per section. There might be tilings, and then you have multiple, but it's a section-oriented way of storing the data. And that's of course great if you just want to have like a look at a single section and browse around that. But if the sections get very large, then you have to load in the entire section into main memory to actually look at that. So that's a technical limitation, which can exhaust resources of your laptop or the workstation you're working with. But it gets even worse if you want to do a cross-section analysis, right? So when you remember, like in Webknossos, we have these 3 different viewports that are actually 3 orthogonal virtual cuts to the data set. And if you want to have a cut along the z-axis, then you need to load in all the sections that are affected by this cutout of that you want to look at. And if you have a section-oriented way of storing the data, then that's very inefficient because you need to load in all these different image files from the sections that are affected, and that can really be too much for your system, and that can be very inefficient. So the thing that OME-Zarr can do differently is that it is not limited in that way that it has to organize the data by sections, but it can actually organize it in an arbitrary way. And the way that Webknossos stores its data is to divide up these 3D or n-dimensional data sets into small little cubes that contain little pieces of the image data. And the nice thing about these cubes is that they can be accessed individually, right? So when Webknossos fetches some data, then it only fetches the tiny cubes that are required to fill up your screen. And there are only so many pixels on your screen, so it's not a ton of data. And that's what makes this interactive streaming and visualization very, very efficient. And this is going through these little cubes is also very efficient if you want to do parallel processing in the cloud or on a high performance computer, because if your data is 3D or multidimensional, then you might also want to do 3D algorithms on top of your data. So then, and if the data is already organized in that way, then that's very efficient for both reading, but also for writing data out. Sorry, I have one more slide that got skipped here, which is this one. So another very important thing about OME-Zarr is, of course, that it's an open standard. So it's not just Webknossos that uses it. And when the data is stored on a server, then it can be accessed by multiple different tools. So here, for example, is NeuroGlancer, which is also another web-based visualization tool which has been developed at Google. Here you can see Mobi, which is a Fiji plugin. And here on this iPad, this is Webknossos. So this, when working with OME-Zarr, then you'll have access to a large number of applications and can support it. And if you put your data on a server, which could be the Webknossos server, then you can work with all these different tools and use the different features that they provide without having to reformat or convert your data for every single tool, right? And this is particularly important if you're working with very large data sets. Imagine you have a data set that is a few terabytes in size. Then would you really want to go through the hassle of reconverting that data set just to open it up in these other different tools? And you might have you have a duplicate of that data set, you need to buy another hard drive. So this is really becomes quite a hassle. And this is really the challenge that we want to overcome with OME-Zarr. It's a standardized file format that a number of different open source tools and others support, so that you don't have to convert your data anymore, and you get the best performance out of it and all of that. So the slide I already showed. So, and then with all of that, this really realizes some of the FAIR principles that was already realized for smaller images, but for large images, it was quite a challenge. But with OME-Zarr and the way of storing it on a server and being able to access it on a streaming basis without needing to reconvert makes this really fly. So just imagine for a second, there's this person over here who acquires some image data and it publishes it and or publishes the paper, but also makes the data available, right? And then there could be another person who accesses that data through a tool like Webknossos or Mobi, and then they do their custom analysis on that, and then they publish it as well. And of course, they cite the work of the author over here. And then there might be somebody else who acquires a similar data set, also publishes that, makes it available, and then there could be another person who only accesses that data, doesn't even acquire it on their own, and now they do a comparative analysis of these two different volumes that have been published, and then of course they publish and cite again. So there's really this virtuous cycle of the FAIR principles, which is findable, accessible, interoperable, and reproducible. Is facilitated through OME-Zarr because you can just access the data. You don't have to download it in full to just look at if that data actually is useful for your project. And then you can still analyze it as if it were your own in your favorite tools, and then use it for your analysis, and then eventually publish all of that. So yeah, this is why OME-Zarr is so great, and all the data in Webknossos is based on OME-Zarr. And another nice thing about that is that it's really an open standard and has support from a large part of the community. Here you can see a paper that we've did 2 years ago, or almost 3 years now. And it has, you might recognize some of the names in the author list here, which are some of the authors who developed some very popular open source image analysis tools as well. All right. So in the next section, I want to talk about some upcoming AI features that we're working on in Webknossos. So, and just for context, my company has been working on doing analysis and in particular segmentation for connectomics for quite a number of years now as a consulting service. And for alignment, for electron microscopy, if you're working with that, then alignment is a very important pre-processing step. You get the data like this, and then you align it so that it looks like this. And that's really key because by doing the alignment, you get a coherent 3D volume, and you can actually make use of the 3D context that this 3D volume provides, right? And if you mess up the alignment, then this 3D information is not really usable. So this is something that usually we spend a lot of time on and these kind of projects. And then once we have alignment done, have a good coherent volume, then another next step would be to run a neuron segmentation on this data set, or some other types of segmentation. I'll have a few more examples in a second. But here you can see an example of this. So this is a fully automated segmentation of a bunch of different neurons in this mouse cortex data set. Yeah, that's something that we'll do on a regular basis. If you are interested in connectomes, then usually the next step after neuron segmentation then is detecting synapses. So that's also something that we have models for that work really well. And then some auxiliary models are sometimes also required for these types of analysis, or for analysis on their own. For example, like nuclei, blood vessel detection, but also mitochondria detection. So this is really something that we've been working on for quite a while now, and now we're in the process of bringing these features into Webknossos so that obviously you can use them directly from the comfort of your browser, right? So it started with alignment, which we've launched sometime last year. So you can upload your raw EM images to Webknossos and then run an alignment from there. And it will run on a server, so you don't have to worry about getting the compute infrastructure right. And it's really just a few clicks in the browser to get this going. And then another feature that we've been working on and started launching as an experimental feature is then automated segmentations. So we started out with a few pre-trained models. So if you have the data in like this like electron microscopy brain tissue, then you can run a neuron segmentation on it. Or if you have some other electron microscopy data, then you can run mitochondria detection and segmentation on this. So these are pre-trained models that you can just run. And again, everything just runs from the browser, and it will run on a number of servers. You don't have to worry about buying GPUs or configuring them and all of that. You just configure like what you want segmented, and then the software will take care of that, and then you'll get an email once the segmentation is done. And then once you have that, you can use all the features that Webknossos has. And now a new feature that we're starting to roll out now is that you can train your own models. This makes this feature much more versatile because you can bring your own training data, right? So the workflow that this will enable is that you upload your raw images, and then you can use the manual annotation features that Webknossos has for generating segmentations, but just in a small bounding box, like you can see here, like a bounding box like this size. Maybe, well, one is usually not enough, but if you give it like a few of these bounding boxes, then you can already start training models from this. So you can teach the model what it is looking for to segment out. Then you can start a model training again from the browser, and it will run on a GPU cluster. And then once the model is done training, then you can use this to roll it out onto your full data set. And once you have done that, then you can use again all the features that Webknossos has to offer for visualizing that data, sharing it with colleagues, doing corrections to the automated segmentations, which will likely be there, and and export it, get some statistics out. So all of these features. And of course, because everything is OME-Zarr, you can also take that data while it is stored directly on Webknossos and export it into other tools where you could do some further analysis of whatever it is that you're particularly interested in. So yeah, so this is something that we're in the process of rolling out now, and we'll have to share more about this in the coming months. So to give you a brief summary of what the Webknossos platform is. So first of all, it's a web-based platform. So everything runs in a web browser. That means you don't need to install anything. And everything that you work with has a URL so that you can easily share it with other people. And then it really enables the workflow for doing different pre-processing steps. You can visualize your data sets of really arbitrary size. Then it allows collaboration over the internet, so really worldwide collaboration. You can annotate your data for analysis purposes, but also for generating a ground truth for segmentation. Can also do automated segmentation with these new AI features, and automated segmentations can then also be proofread inside of Webknossos. And then once you're ready to publish, then Webknossos can also be used to make your data available to the community. So you can put a link to your Webknossos data set directly in your figure caption, and then users will be able to explore your figure interactively on their own terms and start their own analysis on that. And all of that while Webknossos has data management capabilities built in. It works with a variety of different modalities, and through the use of Zarr, it's also interoperable with a number of different tools, and everything is cloud ready. It runs in the cloud, but it can also be installed locally or on institutional high performance clusters if your institution has that. So yeah, this concludes the first part of my presentation. If you want to try out Webknossos on your own, then you can just go to webknossos.org and start working from there. You can upload some data on your own and play around with that. And yeah, so I think we could do a bit of a Q&A session right now, and then I would also be happy to just give you a bit of a demo to show you what Webknossos looks like in practice.
That sounds great. So in the Q&A part, we already have 6 questions. Do you want me to read them for you, or can you see them?
I can see them. I don't know if anybody else can see them, so it's probably best if I read them first,
Yes.
right?
Yes.
Okay. So the first question on the list here is, what are the strongest features that differentiate Webknossos from other data annotation and visualization tools like CVAT and Neuroglancer? It's a great question. I'm not super familiar with CVAT, so I can't really comment on that. For Neuroglancer, the main difference is that while Webknossos and Neuroglancer are both visualization tools, Webknossos also has a lot of annotation features built into it and data management features that are not in the standard version of Neuroglancer. So that's the main difference. But yeah, because everything is OME-Zarr, you can also visualize any data sets that you have in Webknossos and directly stream them to Neuroglancer if there's anything that you're missing in the visualization features of Webknossos, for example. Okay, so the next question is, how is Webknossos addressing the subject of data provenance? Is it possible to version the data as it being acquired? So that's a good question, and there are different ways how you could achieve that in Webknossos. So one is you can upload the data sets to Webknossos and attach basically arbitrary metadata that you would like to it. So if there's like a version number that you want to attach to the image data itself, that's certainly possible. If you want to I don't know, attach any other acquisition specific metadata, then that's also something that you can do. In terms of all the annotations that are generated inside of Webknossos, so if somebody brushes or uses a quick select feature or whatever really, all of that is versioned inside of Webknossos. So there's an undo feature, but also there's a very detailed version log, so you can go back to any version of the annotation in any time. So yeah, and I hope that answers the question. If not, then please let me know, and then I can do another round of this. Okay, excellent. So the next question is, to which extent is the Webknossos data format compliant to the OME-Zarr spec, and what are the differences to the bioformats layout? Okay, so Webknossos supports the OME-Zarr spec to well, we have a pretty wide coverage of the spec itself. I think there's a table somewhere on the website that compares like which different features of OME-Zarr are supported by which application, and so you can go through that list on your own. And but we try to be very compliant with the spec. In fact, we're also a big contributor to the OME-Zarr spec as well. So the bioformats layout, as far as I know, is just a way for for storing multiple images in like a series of images, and so it's basically a container format of multiple images. And when I say images in this context, then I actually mean like a multi-scale image. So I'm not 100% sure if we can import that directly, but all the individual multi-scales should be very easy to import into Webknossos. And in a demo in a few moments, I can show you how you can actually import some OME-Zarr data into Webknossos. All right. Okay, so the next question is, hi, this is a very interesting presentation. Thank you. I have a bunch of questions relative to Webknossos. So the first question is the cost. Is it free to use? And the cost of data storage. So Webknossos at its core is an open source software. So you can install it on your own server if you wanted to, free of charge. There are a few features that are in the paid version. And we do on our website have like a big table that outlines like which different features are available in which version of Webknossos. There's also some costs on that table. And yeah, but probably more importantly, if you go to webknossos.org, then you can create free accounts, and they come with 50 gigabytes of storage. So you can upload your own data, and when you upload it, then of course it's private by default. And then you can start working with Webknossos, and if at some point you run out of storage, then there's a way of upgrading to a paid plan to get more storage. There's also ways of connecting external storage systems. So if you already have your data stored somewhere else on the internet, then you can just import it into Webknossos, and then that doesn't count against your storage quota. So yeah, so there's really, I would point you to our website, and we have all the price information there. Okay, where is the data stored? So yeah, we have a number of different servers. Most of them are in Germany. So that's where they're stored. If you store them on your own servers and just import them into Webknossos, or if you set up your own version of Webknossos, then of course that's where you put it. Yeah, and yeah, we also have managed installations for on-premise. So a number of larger institutions have Webknossos licenses that run directly on their servers, so they can reuse the storage systems that they have already invested in. Okay, and what are the input image formats required? So if you already have OME-Zarr, then that's great. Then you can just use that. If you have a stack of TIFF images, that's also great. So we support a wide variety of image formats. We have also integration for bioformats, which should be able to read a large range of different file formats. So yeah, so if you upload something that is not OME-Zarr, then we'll try and convert it automatically, and then it will be OME-Zarr from there on, and then you can use all the Webknossos features. Yeah, so yeah, I don't know, if you have a TIFF stack, that's probably what I would recommend to then just upload, and then it gets converted automatically. Okay, the next question is, you mentioned Webknossos is using SAM2 for segmentation model. What is your opinion of mobile SAM versus the SAM classic model? I don't think I know enough about mobile SAM to comment on that. There's also micro SAM, which is optimized for microscopy data. I don't know what the current status of that is, but last time I looked, it was based on SAM1. And so, yeah, but we went for SAM2 mainly because it's just worked better than compared to SAM1 on a number of different sample images that we tried it out on, but also it had these object tracking features that we wanted to use for 3D segmentation. So that's really why we're working with SAM2, and it's great. And now there's SAM3, so we're looking at how we could integrate that as well. Okay. Thank you. So next question is, awesome presentation. I admire your work. Thank you very much. For a small facility, how easy would it be to set up a Webknossos server? How much infrastructure would you need? Do you have a deployment kit? That's a great question. So if you know a bit about how to run your own servers, and if you've installed something through a Docker container, Then if you can also go to our documentation, and I can show that in a second. There's a page about how you can set up Webknossos with the open source version on your own, and then run it from there. So in terms of infrastructure, I think like the main thing that you will need is the storage for the data itself. That's really the biggest investment probably if you're working with large images. In terms of the server requirements, I think there's also a section on that open source installation page as well. What are like the minimum requirements? It's not too crazy. So with like a regular server, then you can get going. So it's not too expensive to get that infrastructure to run Webknossos. Okay, so the next question is, I see the demo on 3D EM data. Do you think it is suitable for fluorescence microscopy as well? So certainly, yes. So we have a number of users that use Webknossos with their light microscopy data of all sorts, really. Like when we started out developing Webknossos a decade ago, our primary focus was EM, but we've been trying to extend that focus ever since. So all of the annotation tools work. Like we have support for multiple channels with different colors. You can select the histograms. There is gamma correction and all of these things. So you can use it for data sharing. You can use it for data annotation purposes. That being said, some other tools offer more capabilities that are tailored towards like visualization specifically for fluorescence microscopy that Webknossos might not have at the moment. But yeah, if you see something like that, then please also let me know, and then we're happy to take a look at that if we could add some of these features to Webknossos. Thank you. Okay, so the cost of model training is the next question. Where's the server for training? So that's something, because we're still rolling out these features, that I don't really have the numbers today. So on our website also you can subscribe to our newsletter, and we'll have some updates on how that'll work very, very shortly. The server is probably also going to be the service that we have. So we're aiming for something in Germany, or at least in Europe. Okay. So the next question is a follow-up question to the bioformats plugin. It exports metadata as OME-XML that can be placed in the OME metadata OME-XML file of an OME-Zarr according to the specs. Would Webknossos record an OME-XML using bioformats upon conversion to OME-Zarr and then read the OME-XML when displaying the image and its metadata? That's a very good question, and unfortunately the answer is no. So we don't parse the OME metadata at this point, but I think we could. So that's basically a matter of if enough people ask us about this, then we'll probably implement that. I think so Webknossos has capabilities for storing this metadata, so it would really just be that we need to import it from like the bioformats format, and then put it into the Webknossos metadata, which should work. Yeah. Thank you for that question. Okay, and the last question for now is how about Globus integration? Is there a recommended way to upload/share data in Webknossos? So we don't have a Globus integration right now. It's a browser-based upload, so you can take your data and drag and drop it into the browser, and then it will be uploaded. And there's some resilience built into that. So if your connection drops in between, then individual parts of your file can be basically resumed, and then it will continue to upload. That being said, I recognize that like uploading a few terabytes through the browser is not very pleasant. If you do have that, then please just send us an email. We have FTP servers where you can upload the data to, and then they'll be on Webknossos directly. So that's something that we can provide as part of like the support of our paid Webknossos accounts. Okay. And if you have your own servers, again, then you can upload your data to your own servers and then just import it into Webknossos from there. So that could also work, and then you can use whatever upload tools you have at your disposal, of course. Okay. So thank you very much. If you have any additional questions, then just put them in the Q&A window here. I will be sure to look at them in a few more moments again. And now I wanted to show you what Webknossos interface actually looks like. So here's my demo account. So if you go to webknossos.org and create a free account, then this is the dashboard that you'll get. There will be like a welcome banner on top, and you probably won't have any data sets because you haven't uploaded them yet. But that's the general first screen that you're going to interact with. If you want to upload your own data, then you can press on this blue button here where you can upload some data, and really you take your files and you drag it into this area here. There's some metadata that you need to provide, like a dataset name or where you want to put it in the folders in Webknossos, and then you can upload them. And then if it's already a compatible format like OME-Zarr, then it will be picked up directly. And if it's not, then it will be converted, which is usually also pretty quick, obviously dependent on the data size, but it's fairly quick. And then you'll get an email when it's done, or you just leave open the browser window and it will tell you. So that's the primary way of getting data into Webknossos. There's also this add remote dataset tab on top here, which is if you already have some data that is in OME-Zarr or these other formats, then you can just enter in a URL here, and it will import that, and it won't copy the files, so they will be streamed from the location where the data is actually present. And this works with HTTP servers, but also with S3 and Google Cloud. And so there are a bunch of different places where you could store your data to then load them into Webknossos. So going back to the dashboard, let me show you what that looks like. Here's a sample data set that is actually streamed from IDR. So this is not hosted on Webknossos, but is streamed in. It's a fluorescence data set. You can see here how you can access multiple layers in Webknossos. So you have these two are activated right now. They have different colors, and you can just browse through them, visualize them. There are a few, like the primary controls are listed also in the bottom here. So you can work with that. You can zoom in and out, of course. So there's different interactions that you can do. There's also this data set happens to be segmented already. So you can also work with that. And you can also compute some meshes from these. So that you get 3D visualizations of these segmentations, right? So, oops, that was the one I already had. Here we go. Just a few. Of course, you could click on all of them, and then you have like a nice 3D visualization here. If you want to work with these segments, then you can also go here into this list, and you can also download these meshes if you wanted to, and then load them into Blender or something like that. So that all works. Now let me go to another data set to show you some of the segmentation features. So this is an EM data set, and annotation features. So if you want to create an annotation for any of the data sets you have, then you need to press this button, create annotation. And then you're in this view, and you can see already that there are a bunch of additional tools that appeared up top here. In the presentation, I only really showed you what the segmentation tools look like, but there are a few others as well. So there's the skeleton tool. So skeletons are line graphs. They're great for making like quick annotations of, I don't know, tube-like structures. So for neurons is great, or vascular systems. And let me see. So basically these are like a graph that's connected by multiple dots. You can have branches to them, and you can get a quick visualization of the trajectory of these biological features in this 3D viewport. You can also use them to do some analysis. So for example, if you want to know like the path length of that, then you can get that from here, for example. So there are multiple things that you could use this for, right? You can also organize these into folder hierarchies. And so if you want to annotate multiple neurons, for example, then you can put them into different folders based on cell types. You can change the colors, of course. So there's a lot of things that you can do. You can also use this just to mark out some spots. There's a dedicated mode for that. So every single time you click, you get like a new color. And then you could change the name of these dots here with some comment for, I don't know what it is that you're really looking at here. Yeah. So yeah, that's a skeleton annotation tool, and it's quite versatile. It's also quick to get some annotations in, but yeah, so it's of course limited because it's only these line graphs. Yeah. So another feature is that everything Webknossos is autosaved, right? So you don't have to press this button. It will just automatically save every 30 seconds or so. So your edits are really safe on the server. And if there's an issue with storing the annotations, then this bar up top will turn into a flashing red. So you can't miss it really. Okay, let's jump into the segmentation tools. So the most simple one is just a brush tool, right? And I'm really bad with drawing on a trackpad, but you get the idea, right? I'm sure everybody of you has seen how a brush tool works. Like, there's a dedicated tab for segments as well, and yeah, you can use this. I mean, the brush really provides you as much precision as you need. And let me just fill this part in the middle here. 2D fill. Here we go. Now it's filled in, and yeah, but of course it requires you to go through it manually. As you can see here, I actually skipped a few sections because I wanted to show this interpolation feature. So if you click on this button over here, then it will do a linear interpolation between the two sections that you've been working on, right? So there's no smarts here; it's just linear interpolation, but it can be very efficient if for tracing objects in 3D and going quickly through them. Okay, there's of course also a eraser. There's also a trace tool, which works quite similar to the brush tool, but yeah, you just trace around basically the features. There's a fill tool, which works on the segmentation, so you can fill holes or relabel segments, things like that. There's a pipette tool, which allows you to pick segment IDs, or if you're interested in like the intensities of a particular voxel, then you can also get that. But the next thing I want to show you is this quick select feature. So that's what I also had in the presentation. So the way it works is you drag a box, or is also the point, just the single click interaction, and then it tries and semi-automatically segment out your the feature that is underneath that. You can also click on this button over here to increase the prediction depth, which is this 3D segmentation feature. So let me show you how that works. So now I went to another section, and this will take a bit longer because now it's doing segmentation on a number of different sections in one go. Okay, and here it actually didn't work super well because I think this, yeah, it's a bit hard to see, but I think the segment stops here or morphs into this. No, it doesn't morph into this other one. So yeah, this is a tricky thing where then, of course, again, you can move to your brush tools or your eraser tools to do any corrections. In the interest of time, I'll leave this as it is right now. So let me pick another one, maybe that works better. So let's just go over here and so pick a new color here. Okay, and now it actually has drawn and also stopped because the segment stops here. Yeah, the predictive was quite a bit more, but because it actually did the right thing here and stopped, which is great. Yeah, so again here, when you're drawing, you can also load these meshes. This one is probably super tiny now because it's just a tiny bit, but yeah, it's also, I just did like a single click here to annotate that, and yeah, and if you continue drawing, then the segments will also update, and then you can yeah, it will grow and everything. Yeah. And there's also a measurement tool. So if you want to get some quick measurements for some diameters or something like that, then you can do that. Or yeah, so these are not persisted. So it's just for getting quick ad hoc measurements out. Yeah, and another annotation tool that Webknossos has is this bounding box tool, which is it's really just for drawing some boxes in your data, and that can be useful for a number of different reasons, right? So first of all, if you were thinking about acquiring ground truth for your segmentation model training, then you can just go through your data set and drop in a few different boxes, and then you can use you just label all of these and then feed that as training data into your model. And of course, because everything runs in a browser, right, you can get multiple people to help you with that. There's also a task system to divide up the work and manage all of that if you're doing larger projects. I won't go into that into detail today, but here's a number of different sections in the docs as well on that to facilitate that. So that's one use for these bounding boxes. Another use is to actually download some data. So you can go to this bounding boxes tab over here and then click on export data, and there you have the ability to basically download this piece of data as a TIFF or OME-TIFF in that case. So yeah, you can pick whatever layer you want, and so for example, if I go for this volume, then that would be the segments that I've just drawn, and then you can download that, and that should it will prepare the TIFF files on the server, make them available for download. It takes a few seconds for this size of bounding box. If you go for larger, then it might take a few more seconds, and then you can download that, and then you can open it up in Fiji or any other tool really that you want to. So yeah, so that's another great use of using bounding boxes in Webknossos. Okay. So other features that are in here are, of course, everything's not only autosave, but you also have undo and redo if you need to, if something went wrong, and then you can always go back. And as I mentioned, there's also a full version history. So from the very first edit that you do, you can always go back in time and fix any things that went wrong. If you're interested in trying out the AI analysis tools, then this button over here is what is important. We after doing the slides, we have redesigned that quite a bit. So yeah, here you can basically pick like the models that are pre-trained and are available, and then you can run that on your data set. If you want to try out the alignment, then you can pick this tab over here and can select to align your data set, and this will be the place where you can then train your own models on from Webknossos directly. So as I said, this is something we're rolling out at the moment and over the course of the next few weeks. So feel free to keep an eye on that. Okay, if you've ever want to try out Webknossos on your own and have any questions, then there's also a help button over here. So you can, it's basically a quick way to send us an email, and then we'll be happy to help you out and hopefully solve the issue they may have, and as quickly as possible. Another place to get help is in this help icon here, and there's user documentation. So let me show you. There are docs for all the different features that Webknossos has, and many of them have videos attached to that. So here's just an overview in 100 seconds. But if you go to this getting started guide, then there's a video it's like 15 minutes that it goes through the main features of Webknossos a bit more in detail. So that's a resource that you can use. And another thing that you can do is go here, which is basically just a link to the Image SC forum, where we're also on. So if you have any questions, then you can also ask them there. And that's great because then other people who might have the same question can also see it and participate in the conversation as well. Yeah, and there are a few other ways how you could also interact with us as well. Okay, so yeah, one more feature that I want to show you is this feature called Zarr links. So that's a great way for creating basically to use the data that is stored on Webknossos in other tools. So I just created a Zarr link, and So let me go to NeuroGlancer right now, just as a demo, how that could look like. So you can take the data that is here, and I'm just going to take the EM data right now. I'm going to tell that it's Zarr, and I'm going to load it in. And then you can see that the data is here as well. And this is now streamed directly from the Webknossos server. And maybe I'll also, just to show you, also add the segmentation that I've drawn, because you can also add those. Zarr again, like this, and it's not an image layer, but a segmentation layer. Okay, now it's here, but it's mostly empty. So now we need to find actually the location so that we can see something. Here we go, and here you can see, so this is segmentation now popped up in NeuroGlancer as well. So that's just just to illustrate that you can go quite easily between tools, and all of that is due to the power of OME-Zarr, of course. And yeah, and you can really use Webknossos as a place where you store the data and do your annotations, and then if you need to go into another tool, then you can just stream the data out. Okay, just before we go back to the Q&A, just another plug for the Python library as well. So if you know your way around Python, then this Python package is a really great way for automating all things around Webknossos. So you can, again, use it to upload some data sets, or you can download image data from that. Yeah, so there are a bunch of different examples that you can use, and basically this library is a thin layer around all of this, around OME-Zarr, and adds authentication on top so that you can securely access private data that is stored on Webknossos. All right, okay, so now I'll go back to the Q&A. There are 2 questions in here. All right. So the first question is, I would be interested in trying out the import/streaming in from IDR with a dataset from the IDR in my own Webknossos account. Does a how to exist that shows the needed steps? Okay, and then you answered yourself. I think I found the answer to my question here, and then here. Yeah, so that's pretty much it. Yeah, I mean, I can also show you how it looks like. Just need to find the right URL here. So because it's basically what I did with this live microscopy data set that I just showed you. Let me just find it. So here it is. I can click on this thing, and it gives you access to like the different URLs that you can access data from. So let's just pick the first. I think that should work. And I can add the data set, remote data set right here. I press add layer, and then we'll fetch the metadata. I need to give it a new name. I don't know, let's just call it like this. Then you can review that all the metadata is correct, right? All the different layers got picked up, and then let's import that. And yeah, then you should have access to the data. It also imports some of the rendering metadata, right? So that the histograms are proportioned correctly and colors are set. So yeah, and also the labels are here. So that's basically how you import a data set from IDR. Okay. So I guess if there are no more questions in the Q&A, then I think I'd like to again thank you all for coming and for your attention. If you do want to try out Webknossos, then please do that. Just go to webknossos.org, and that's really the entry point. You can also find us on social media, of course, or just send us emails if you want to. And yeah, thanks a lot.
Thank you.